research project. My name is James Randiak Alaro. I've actually dropped James. Now it's just Randiak Alaro. Hopefully we are going to have a great discussion here. I'm from the Center for Global Health at the National Cancer Institute here in the US, and I'm a scientific program person in one of the projects in DSI Africa. We are going to have two discussions, two talks in this group: the genome-wide characterization of complex variants and their phenotypic effects in African populations, and then characterizing effects of air quality in maternal, newborn, and child health. I think we have, because we only have two talks, we have about 40 minutes. So I think if we make our talks somewhere about 10 minutes, 12 minutes, it will allow sufficient questions for each group. If you have questions, at the end, you may ask those questions by raising your hand. Alternatively, you can also type them in the chat and we look at them as the presenters are talking. So we are going to start with the first talk, which is by genome-wide characterization of complex variants and their phenotypic effects in African populations. So please proceed. You may share your slides. And before, again, as you go ahead, please feel free to introduce yourself.
Okay, just a minute. Let me try to get my slides. I'm sorry, I'm just
no problem. Just get it done. All right.
Yes, hello?
Hello?
Yeah.
Okay.
Okay, so just introduce yourself and you may start again. Keep within the 10 to 12 minutes to allow people sufficient time to ask you questions. Are you good?
Yes.
Yeah.
So let me quickly go first. Hello,
Just
everyone.
in presentation mode, if you will. So it's,
yes,
it's
yes. It's coming. Okay,
Great.
so hello, everyone. Good afternoon from here in Nigeria. My name is Itunu, and I'm going to be presenting on behalf of the NPIs for the project called Genome-Wide Characterization of Complex Variants and their Phenotypic Effects in African Populations. This project was newly awarded last year. We joined the DSI just about a year ago. I'm going to be presenting like an overview of what the project is about, and I will give a few highlights of what we have achieved in the 1 year that we started, and then a few points, on the next line of action for us as a project. This project originally came from the H3 Africa Genome Analysis Working Group, and it's a group of 4 universities. The host universities are Covenant University in Nigeria. We have Makerere University. We have University of Cape Town also contributing to this project, and we have a US collaborator, University of California, San Diego, also on this project. And I'll be presenting on behalf of the PIs today. So this is the presentation outline. If you don't know about tandem repeats, let me just quickly tell you briefly. So tandem repeats basically are like repetitions of sequences that are paired together. They have to be appearing consecutively. We have different types of tandem repeats. We have short tandem repeats and we have variable number tandem repeats. So on the screen here, you see a little sample of what this tandem repeats look like. The tandem repeats are short tandem repeats are typically shorter, and the variable number tandem repeats are variable from 6 base pairs and above. Sorry. I'm sorry about that. I don't know what went wrong. Let me check. I'm sorry, there's something wrong with the slides. Let me pick it from the Google Drive directly one more time. I think the downloaded one is having some challenges.
Yeah, that is okay. Let's get it. Might be easy also if you convert to a PDF.
PDF media. Because I have it in the Google Drive.
So you can just download it and then convert in PDF.
I downloaded already, but the what's it called? The fonts are looking funny. Okay, please, can I just ask the next person to present, and then let me just quickly get this sorted. I'm sorry.
Yeah, we do that. We can do that if the person is ready. So this is characterizing effects of air quality in maternal, newborn, and child health project, and I think is by Dr. Makanga.
Yes, that's correct.
So Dr. Makanga, just a quick introduction of yourself, and again, we'll try to get there about 12 minutes with your presentation to give people about 5 minutes or so to ask questions.
Okay, thank you. Just confirm if everyone can see my screen.
Yeah, put them in presentation mode.
Okay.
Perfect. Mm-hmm.
Good. Thank you. And thanks to the organizers of this meeting for the opportunity to present some of our work. Again, similar to the previous presenter, ours is a UO1 that was awarded in the last quarter of last year. We've been gaining momentum, and I would say we've got a full team now in place. Yeah, so listed below are the partners that are part of our work, some of them more closely and directly, but some maybe a bit indirect. Our project is called Characterizing Effects of Air Quality in Maternal, Newborn, and Child Health. I'm one of the MPIs on the project. We've got Dr. Tamara Govindasamy, who's also the other PI who's on the call. Maybe, so I'd prepared a 6-minute presentation, but I think I'll flesh things out a little bit more since we've got a little bit of time. The aim of the project is essentially 5-pronged. And I'll further unpack more aims, more substance to the aims in the next slide. The first aim of this work as a supplementary grant is to advance capacity for the application of data science in the field of environmental effects, and specifically looking at ambient air pollution and its effect on health, focusing specifically on early career researchers and building capacity for these with at an institution in sub-Saharan Africa. The more scientific aims are probably 2 to 5. As part of our work, we aim to design air pollution spatial indicators and proxy measures and validate these using ground sensor data. Many of us are aware that on the African continent, we've got a very poor network of ground sensors for measuring air quality. So the aim of this work is to see if we can design other alternatives in addition to the ground sensors that accurately predict exposure to poor air quality. The third aim is to say, okay, once we've designed these proxy indicators, to make them widely available for reuse to a broader audience who are interested in investigating the impacts of air quality on health outcomes. Primarily, our primary focus is maternal, newborn, and child health, but I think the proxies could be used for scientific purposes in pursuit for other health, for understanding the impacts on other health outcomes. So this is creating sort of a federated framework of data, of access to data where people can reuse some of the data that will come out of this project. Our fourth aim is to essentially explore the relationship between exposure to poor air quality and the outcomes of pregnant women and their babies in sub-Saharan Africa. So this forms more of the scientific aim, or more of the epidemiological aim of the study, where we'll explore these associations, and also considering other factors that may act as confounders to this association. And ultimately, the idea is to translate evidence from this study into tools that can be used for intervention, inform policy, and ultimately achieve impact in reducing the effects of air quality, of poor air quality on health outcomes. Because air pollution is currently the second leading risk factor for death in the world. So as part of this work, we are leveraging the work of the HEAT Center. Those who would have attended the previous session in room 1, we had the HEAT Center presenting much of their work there. And the HEAT Center, it's an acronym that stands for Heat Health Africa Transdisciplinary Center. We're mainly leveraging Research Project 1. Chris would have spoken to both projects. We're mainly leveraging Research Project 1 in terms of the data that's been collected about, I think now it's an estimate of around 2 million individual health records. The climate variables that are being assembled primarily by our partners in Cape Town, and also the thinking around early warning systems within the project, which feeds to our last objective, number 5. As part of RP2, which is also looking at heat indices, we intend to have some thematic learning from them and perhaps transfer some of what happens in RP2 into defining some of our exposures as part of Chiki. I think of importance, we're bringing in a new partner to the consortium, which is the Midland State University, where we currently have a postdoctoral data scientist and a PhD or potential PhD student who will be looking at characterizing exposure, validating these pollution proxies, as well as performing a good chunk of our analysis. As you can see, our team is quite balanced. We've got 4 women and 4 men who are part of the core team. Our extended team is, would like to assume, equally balanced. Professor Stanley Lushters and Dr. Makanya are both the core PIs of the HEAT Center, so they're coming in to support the project and help integration with the HEAT Center. So in terms of timeline, we're coming towards the end of year 1, but a good chunk of the first half of the year was largely contracting and, setting up some of the base infrastructure that we'll use going forward. And I would say maybe in the last 5 months or so, that's when we've sort of started getting our hands dirty with looking at data and making progress to some of our objectives. So year 1, we've aimed to do a literature review, collect some of the core data sets that we're intending to use, make strides towards validation of existing proxies and maybe new proxies that we'll design as part of the project, and do a lot of data pre-processing in preparation for much of the crunch work that will happen in year 2. So in year 2, we're looking at taking the exposure data and cross-referencing that with health outcomes that are coming primarily from research project 1 of the HEAT Center. We're looking at exploring different methods and also furthering development of new indicators depending on the new knowledge that comes out from our analysis. So year 2 is where much of the work will be done, and we're hoping that year 3 will have some scientific findings to push out through different channels, including publications, but also perhaps policy briefs. And we hope that our 2 PhD students would have made advances on their PhDs. The general framework of our work. So because of sort of low density of sensor data, ground sensor data, we're looking at deriving air quality proxies from primarily satellite images, but are also planning to use geospatial data sets to develop some of these proxies. And I think I'll speak to 1 or 2 of the proxies in the later slides. We're interested, again, as I mentioned, in health outcomes that are being collected across the continent as part of the research project 1 of the HEAT Center. We've got access to some good quality personal exposure data that was collected as part of the PRECISE network. If you were in room 1 in the first session, you would have had Liberty talking about this personal exposure data that he collected. But I think in addition to that, Laura, who's also part of our team, is starting to lead sort of the process of systematically gathering sensor data from different studies or sources across the continent. And the idea is to create continent-wide high-resolution pollution exposure maps that will cross-reference with our health outcomes, and then quantify associations with maternal, newborn, and child health. So what have we done to date? Much of our team is they're actively taking courses on machine learning. We've started the process of validating some proxy indicators that had been developed as part of a previous project. So normalized difference vegetation index, distance to highways, which is a measure or a proxy of traffic-related pollution, altitude, and how the air moves differently in different altitudes, and perhaps inducing different exposures in different places. The quasi-direct air quality measures for PM2.5, NO2 that are satellite derived, and road network density, which is also a proxy for traffic-related air pollution. So these are the proxy indicators. The personal exposure data that I mentioned earlier, this is data that was collected in Mozambique, the Gambia, and Kenya. And we've got about just over 1 million data points captured from 343 women who carried personal exposure monitors throughout the year in the 3 countries. And so far, based on pre-processing, we've managed to quantify PM2.5 exposure and NO2 exposure reliably as the air quality exposures of interest in this study. We've got a few ones that are still being validated, PM10 and I think SO2, that we could still use as part of this work. So Bongani, who's sort of taking a lead on this, has started cross-referencing our proxies for road network density with values for PM2.5 and NO2. It's still early days, and at this stage, I do not have quantitative results to show. In terms of progress, we're also, as I mentioned, starting the process of systematically collecting data across the continent on air quality indicators. We've got our data management plan that we're hoping to finalize by the end of the year hopefully by the time we have the Climate Health Africa Conference in Zimbabwe, we'll have an opportunity to just revisit our finalized data management plan, or our data management plan, and finalize it at that stage. We've got a few abstracts that are being submitted to DSI concerning this work. In terms of publications, we've got a systematic review that's underway, looking at the effects of combined pollution exposures on preterm births, as well as a protocol paper describing objectives and methodology for our study. So I hope this is useful to those that are interested in this, and we're really looking forward to feedback and opportunities to collaborate. Thank you.
No, thank you very much, Dr. Makanga. Any questions from the audience for Dr. Makanga's program? And you can just stop share so that I can see people. Judith, I see your hand up.
Oh, hi, yes. I work at Fogarty International Center in the communication department, but I just had a question. Are you going to be cross-referencing anything about indoor air pollution, you know, from traditional cooking or anything, you know, with the outdoor air pollution data?
Excellent. Thank you, Judith, for your question. So yes, we're also considering indoor air pollution as part of the PRECISE study with the personal exposure monitors, where we collected data in the 3 countries. These personal exposure monitors were worn both outdoors and indoors. So we are currently trying to segment the data, and our validation process so far is focused on outdoor air pollution, but we've got a chance to take a closer look at indoor air pollution. It's a little bit harder to segment and actually identify which instances are indoor compared to outdoor. So that's why we started with outdoor from our personal exposure data. But I think as a next step, we're going to be working with indoor data as well.
Thank you.
Thanks.
Yeah, as we wait for the next question, I'm curious whether you are aware of the GeoHealth program that is again coming from Fogarty, funded through Fogarty. There's a group, Dr. Kiros Behane, I think his name is Behane, Dr. Kiros Behane, from Columbia University, doing his work both in Kenya, Uganda, and Ethiopia. And I think you guys are working in the same space. It would really be great to either make a connection or have a connection between you guys going on. So if you don't mind contacting me after this, perhaps you can.
Okay. I think, yeah, so thank you for that. I think we met him at the last DSI conference in Rwanda. And he's also involved in the HEAT Center.
Yes.
I think as part of scientific advisory. So we've had a chance to link up with him, but we haven't really connected from a science perspective. But I think I agree with you. You'll be a good resource to connect with. Thank you.
Yeah, I will connect you directly. I'm a scientific officer in that program as well, and I can see how you guys overlap. I'll put my email on the chat here for your purposes.
Okay.
And are you, when you look at air pollution, especially outdoor air pollution and movement, people moving from one area to another in the locations that you are, is that a concern or not at all? People moving from one and relocating from one location to another at various points in time?
Yes, I think that certainly poses a challenge when we try to assign exposures to locations, because for what we're seeing in some of our studies, we don't really have good detail about migration patterns. In many instances, we get the woman's home address, but she probably spends a lot of time elsewhere other than her home. And
Right.
in many instances, we can get her home address close to the time when she delivered, but she could have lived somewhere else when she was pregnant. So that's certainly a challenge. Fortunately, within the precise data set that I've given as an example, that's addressed because women come to health facilities 3 times, and from start to end of pregnancy, you can get a sense of their movement, even though it's not necessarily entirely complete. But for many studies, that will be a problem because we only have a single location. And in some instances, it's actually just the location of the health facility where she delivered.
Right.
And
Right.
we've got no real information about where she would have moved. So that's certainly a challenge. And if there are any people with solutions to that, that would be welcome, or suggestions as well. Yeah.
And maybe that is a concept, being aware of that, because you can't also make people not move, right?
Yeah. Yeah.
Just adjusting your studies and your metrics to kind of account for that, I think would be great. I see from Emil Shimusa offering possibilities to collaborate. I don't know whether she wants to say something about this. Emil, you want to elaborate a little bit more, but your email is there on the chat. So there's a question from the audience here. Did you monitor how often they are carrying their monitors?
Yes, so good question. So these monitors, I think battery life was about 5 days. Liberty, you can correct me if I'm wrong there. It was about 5 days, and we're recording a data point every minute for the 5 days. So in terms of so we've got a good temporal sort of spacing for the data. I think the question is trying to get to whether we know what we're measuring is when the woman was carrying the bag, the sensor bag, or not, because she could have left the bag at home and she would have traveled. So for the most part, I would say we don't know whether she was carrying the bag, but that's also of less interest to us because our idea was to just characterize pollution in the geographical region.
Right.
And then assign exposures and then create models that would assign exposures to women that are actually not wouldn't carry the sensor, but who we have outcomes for. So yeah, but we're confident for the most part through our community engagement efforts that they would carry the bags around, and by the geographic diversity in our data, that the bags were indeed moving.
No, great. Thank you very much. Really a great presentation and very much on time. So, Dr. Isawan, are you ready to give it another try?
Absolutely.
Okay, go ahead,
Yep.
please.
Okay. So can you see my slides now?
Presentation mode?
Yeah. So apologies for that. I'm just going to start again. My name is Itunu, and I'm presenting on behalf of the PIs. We are a new project about a year old in the DSI Africa Consortium. The project is tied to genome-wide characterization of complex variants and their phenotypic effects in African populations. This project is a collaboration of 4 universities from Covenant University in Nigeria, Makerere in Uganda, UCT South Africa, and University of California, San Diego. We're all previously members of the genome analysis working group of the H3 Africa before we came together to submit this grant, which was eventually awarded. Here's the presentation outline. Tandem repeats, like I explained earlier, really are patterns of identical DNA segments that are following each other consecutively. They are repetitions and they are directly adjacent to one another. So we have different types. There are short tandem repeats and there are variable number tandem repeats, like you can see in the picture. And a couple of them have been associated with diseases. And so why are we studying tandem repeats? So tandem repeats are one of the most popular complex variants, and they represent some of the most polymorphic regions within the genome. That means they change a lot. And so it's very interesting, to study, what's really going on in that region. And like I said before, they have been implicated in a couple of diseases. For example, we have some neurological diseases, mostly like the Huntington's disease, Fragile X syndrome, autism spectrum disorder, and a few other ones. These tandem repeat variants have been associated with complex traits, also including things like gene expression. So the current studies we have not really been able to explore these tandem repeats within the African population, to a large extent. And so this project really proposing a deep characterization of African-specific tandem repeat variations. And we are looking to review and identify novel tandem repeat expansions and other variants that may be contributing to African disease associations. So what are the specific aims of this project? So the overarching aim for us really is to investigate and identify novel tandem repeat expansions within the population, within African populations, and we are trying to see what their contributions are to different African phenotypes. We have three specific aims. The first one is to perform deep characterization of genetic variants at repetitive and structurally complex regions. We are also looking to apply machine learning methods to detect African-specific signatures of selection at repeat variants. And we're also looking to identify genotype-phenotype relationships and integrate this tandem repeats into calculating polygenic risk scores. So the idea is that eventually, applicability or clinical usage, we are hoping that if we're able to, then we can also propose them as a possibility to include them in calculation of PRS. You know that PRS is currently being calculated using single nucleotide polymorphisms, and these are not the only types of genetic variations that exist. So hopefully, if you're able to do this project and successfully annotate the tandem repeats within the African genomes, the long-term effect of that is that we can now begin to move closer to a point where we can now calculate PRS using this complex variants, not just using SNPs. Yes, so here is the little architecture of the overview of what our project looks like for the three aims. The first aim is just general genotyping and characterizing this tandem repeats, and these are the data sets that we're using. We have secured collaboration and approval for these data sets, and this aim is currently ongoing. We are using different genotyping tools. I'm not going to go too much into the methodology, but different genotyping tools, including an ensemble TR tool, which is assembling 4 or 5 TR genotyping methods and getting a consensus approach. And then in the second aim, we are then looking to select those that are population specific, and we're also looking to validate that experimentally. And after that, we then look at association with phenotypes and then begin to drive towards having a data portal where we can now have an extensive data resource that gives the entire tandem repeats signature in different populations and their summary statistics, and then move towards using that, for other phenotype genotype associations, and then like in the PRS calculation that I mentioned earlier. Yes, so in terms of further breakdown, we have a few projects within our bigger project. And the first project, which we already did and completed, we used the Human Genome Reference 38 for the first round. And then we used this ensemble TR method I told you, where we are assembling different genotyping, TR genotyping tools. This has been completed. Now we are using the telomere to telomere data, for the alignment rather than the Human Genome Reference 38. And we are identifying new tandem repeats. And then after which we are also looking to use the pangenome data set also for the same process. For the pangenome data sets, we will eventually be developing new methods or to modify the HIPSTA, one of the existing methods, to be able to work with the pangenome, which does not already exist. In terms of the second project, this is just basically looking for population-specific tandem repeats within the African population, and then the third one here. For the first one, the second project here, we are using the AwiGen data set, the whole genome sequence data set, which has about 100 samples. And then in order to increase our sample size in the third sub-project, we'll be using the AwiGen SNP data, where we'll be doing massive imputation, and then we'll then be calling tandem repeats after imputation. So the idea of this is just to increase sample size, because we know that whole genome data sets are not available in the kind of sample size that we expect. And so we are going to be complementing the whole genome data set with the SNP data set here in this third sub-project. And then future works here will then be combining the tandem repeats that we have found from the whole genome sequences and then from the SNP data set, together to have like a massive TR calling set of tandem repeats within our population, and then we move on to do some fine mapping. We're hoping to also use the all-of-us data for some meta-analysis and all of that. So because of time, I'll quickly move on to the project highlights, some of our progresses we have made in the last 1 year. So here is one paper that we had already published which was successful in identifying 2 African-specific trinucleotide tandem repeat expansions. So with this data set, we used joint calling from 1,000 genomes and the H3 Africa data sets, the H3A-BILO data. So with this one, we were able to have one of the first publicly available tandem repeat call set up to date. It has about 1.7 million tandem repeats. Some of them have been experimentally validated, and we have 98% confidence. And the first SNP plus tandem repeat haplotype panel was also gotten from this. In order to just show some collaborations with what we already know, we're looking at the PCs here with the 1,000 genome, the African data set in the 1,000 genome, and then also the H3 Africa data set. And we're looking at the tandem repeat calls, and we see that they actually map nicely, and the PCs are really similar to the population substructure that we already know using SNP data. So this is also giving us some kind of confidence in the results that we have, because they are kind of in sync using using the SNP data set, the population structure that we're seeing, and that we're seeing now using tandem repeat calls, are similar, and that also sort of just boosts confidence that what we're doing is in the right direction. In terms of results, I was talking about the African-specific tandem repeats, and we found CTT repeats, within this 2 regions here, and these have been shown to be within our data set to be specific to the African population when compared against the UK Biobank data. A second manuscript is currently in the final stage. We have written in Nature Protocols, submitted to Nature Protocols, but we have written a tutorial guide paper, which is basically just a step-by-step tutorial on how to do the tandem repeat analysis, the entire workflow from start to finish. I believe it's going to be a very useful resource. It has codes, it has data sets that you can use, if you are trying to learn, all of this pipeline from the beginning to the end. Another highlight for us is that we held the first human genome repeat expansion workshop a few weeks ago, which was in May of this year. It was a 2 weeks intensive course. It's run for 10 days, that's every day, every working day, 2 weeks, and we had about 5 hours every day. We had a good spread across Africa. There were about 30 participants in this workshop. It was very exciting, and we had very massive, I would say very massive interest. So we're hoping to run this workshop, subsequently. There'll be several iterations of this, but for the first outing, it was good. Eventually, there were 17 successful trainees that finished, from start to finish. So we delivered this training using lectures. We had hands-on practicals every day. We had research talks And we also had group projects where people were grouped, I think 3 or 4 small groups, not too large groups. And at the end of the training, they were able to present their projects and they were graded. And at the end, 17 people finished, which we're very happy. We had more than 50% success rate, despite the network challenges and work schedule of people, several reasons. But it was really a very good outing, and we were super proud of that. The curriculum, we have a GitHub account, and we had created several platforms where all the materials and everything have been put for further use. But we definitely are going to have several iterations of this workshop, just to continue to train and to develop a cohort of people within Africa that are able to do the tandem repeat analysis. So here, just a list of the MPIs in terms of acknowledgement. Thank you to all of them. We have Professor Iladi from Covenant University, Professor Jim Rick from UCSD, Dr. Jingo from Makerere University, and also Professor Ezekiel Adebi. And then also for the co-investigators, we have Dr. Neil from UCT, myself from Covenant, Dr. Adam also from Covenant, Ibra from Makerere, Young, and Ilya. And then a few of our trainees also have been, really impacted, and they are doing very, very well. I'll be waiting for your questions. Thank you for your attention.
No, thank you very much, Dr. Esawan. Questions for Dr. Esawan? Great presentation again. And so you can maybe stop share so that we can the group. Yeah, great.
Yeah.
Mm-hmm.
Can I ask my question?
Yes, please.
Yeah. Thank you, Dr. Ice1, or I don't know the name. I'm sorry. Yeah, you did a great presentation. It was very interesting. But I have some questions. I don't know if you can allow me even to ask 2 or 3 questions, because your topic was very interesting. So I want to know, when you're doing the multi-populations, the GWAS that we are going to do, is it, are you going to do or meta-analysis GWAS, or is just normal GWAS specific for different populations? Because if you are doing GWAS meta-analysis, how did you deal with different genetic background of different populations in Africa? Because we are in Africa, but we know that different population came from different background. How did you deal with meta-analysis? The second question is about how did you identify your...
Yeah, can you just let her answer maybe one question at a time so that she doesn't get lost? Then we'll give you an opportunity to answer the next.
Okay.
Or they are related. If they're related, it's fine. You can go ahead.
Yeah, should I ask this second one?
Yeah, go ahead. I wrote the first one down already.
Yeah, the second one is about the PRS, because you're talking about PRS. It's a good subject, but I want to know, did you try to check the irritability of your population before you analyze your PRS? Because sometimes you have always the challenge of using PRS if you don't know how much genetic, if you don't know how much is your irritability, because you don't just do the PRS because you want to do the PRS. Sometimes it will not explain correctly the different polygenic score for different for different individuals. So my question is, do you know the irritability before you analyze your PRS? If it's the case, is it the is it the irritability for each populations, or you do the irritability for overall the population for Africa? If it's the case, did you understand the different background of those populations? That one's
thank you, Dr. Kabongo. And Dr. Esawan, I think Pedro Fernandez is also maybe asking a related question here about whether the consortium proposes that there will be a robust PRS for the whole continent for specific condition, or would there be regional PRSs?
Yeah, it's a major question, because you can't just do PRS. Because
so I know that the PRS concept is very interesting, and I know it's going to generate a lot of questions, but we are not even there yet. So I was just saying that this is supposed to be like a step towards that goal of eventually being able to calculate PRS using complex variants. Right now, we are not there yet. It's still something that is conceptual. Methods are already being developed, and I'm just mentioning that this is like a direction, that we will be looking at in the future. But the first thing to do is to even characterize and genotype these tandem repeats within our populations first. If you look and you check, you may not find too many studies that are doing tandem repeats in Africa. So this is like the first attempt at doing that. We are a little bit far from using tandem repeats as benchmark or platform for PRS. We will get there eventually, but I'm just saying that PRS calculation eventually will move towards complex variants and not only using SNPs. So I don't want to go too much into that.
Yeah, I was highly interested for that. That PRS, are you going to calculate the effect of PRS for the tandem or just for the other variant? Which card is your variant?
So eventually we'll get there, but what we should want to establish now is to even have like a complete resource where we'll see massive tandem repeats identification. And according to your question for the meta-analysis, first of all, we are starting with populations that are similar and that we already know their population substructure, right? And then we are very, very much conversant of the population and the heritability within this population before combining and pulling them together.
Because when I see your
so if I can hear what you're saying, I think is that PRS is still in the future, and these concerns that have been raised will be considered as you develop towards PRS, right?
Absolutely.
Okay, we can even talk later, no problem, because I have many questions.
Thank you. Yes, yeah. So if you catch me anytime, I will be at the constitution meeting in Mauritius. I'll be happy to talk to you about this.
And you can also connect over the email. I think, Dr. Kabongo, this seems to be an area of interest to yourself, and you and Dr. Isewan can perhaps connect.
Yeah, I'm in Canada now, so I don't know. I can send my email.
Oh, great. Any other question for the group? We still have a few minutes.
Yeah, my email is there. I'll send the email. Yeah.
And again, hearing no other question, I think, Dr. Kabongo, if you have other questions that you want to explore, please go ahead. We have time for that.
Yeah, the other question also was about, I don't know, you talked about machine learning, right?
Yes.
Yes. So which kind of model are you going to apply using your machine learning? So which kind of machine learning are you going to use, supervising learning or unsupervising learning, or which aspect of machine learning, because it's quite robust, your project? Yeah.
Yes. So one of the first points where we applied the machine learning was in assembling. I mentioned somewhere in one of the methods where we had combined different genotyping methods, and we tried to create an ensemble or to find a consensus, among the results of the existing ones, just to sort of increase power and to also be sure that we are not misclassifying them. So it was an ensemble method, and it's in the published Nature Communications paper. It's a classification supervised learning, yes.
Okay, you are doing classification. Yeah. That's good. Yeah, that's good.
I think really genetic works lends to a great opportunity for collaboration across the continent. I think given the contextual genetic diversities and all that. So perhaps Dr. Isewan, this is a space. I know Pedro, for example, has worked quite a bit in this space. Kabongo seems to be interested in this space. This is really a space where we can do different and not just the typical. So if you get an opportunity to really expand your work to be very informative, very early on as you're setting it up, I think there's a great opportunity there. So I don't know whether you want to comment about that.
I think this project is very good because me, I was working more in GWAS. Yeah, I'm more familiar with GWAS and variant analysis. Currently, I'm working something related to the TB. We are trying to analyze the impact of different genome, but using whole genome sequencing, because the previous analysis they are working in tandem. That's why I was very interested for this topic. So it's just my first time to see that we can use tandem to apply, even using GWAS, PRS, and other stuff. For me, I was using GWAS just for the SNP, to identify the SNP, which can be associated with the disease. It was just for the SNP, not for the tandem. So now I get that it's very important.
Yeah. So the idea for us is we are actually doing a computation within GWAS SNP data and trying to see if we can use that to increase sample size, because ordinarily tandem repeats only you can get them only from whole genome sequencing, but many of the whole genome sequence data sets are very small. So with the SNP data, you can do imputation. Of course, the accuracy will be a little bit skewed, but it's still something, rather than just throwing all of that GWAS data away.
Yeah, but the things with GWAS, sometimes have a problem of missing irritability. Sometimes we always have this kind of issues, and knowing that GWAS is based on common variant, sometimes we are not able to capture some
Complex variant.
Yeah, for the complex variant like Ray variant and O variant. So sometimes GWAS is quite limited with this kind of analysis, but it's good what you are doing. So.
And Pedro says the STR angle is unique. So perhaps, Dr. Esawan, if you can share your contacts, I'm sure that from both Dr. Kabongo, Pedro, you guys can talk to each other and have similar discussions around this area. I agree that we have a great opportunity to really inform genome science through Africa. So again, we are—
Thank you for having me. Thank you for
in the last minute for this. Thank you both for great presentations and being timely. Thank you for the participants for being here. Again, these are intended to be very collaborative. Reach out to the speakers if you want, or reach out amongst yourself to make great of this opportunity. Thank you all. See you on the other side.
Thank you.